So I'd like to actually move on to another short talk about DNA databases and investigation. I've shown you this before, but I want to show it to you. I'm not going to repeat everything on here, but I want to show you what this has to do with DNA databases. Because a large part of investigations, particularly with cold cases, has to do with using a DNA database, right? You've all used CODIS, and it works sometimes. It doesn't work all the time, right? Now, the major problem with CODIS is that your evidence isn't always getting up onto CODIS. And if the evidence never makes it onto CODIS, you're never going to be able to find an association with someone who's on the database with evidence that's not on the database. I'm going to explain why and how that all works. Again, in the old days, actually, this whole model was created by the Brits in the UK, the Forensic Science Service, over 20 years ago. And their DNA databases were only single source. They really had DNA from a DNA that only had one contributor. There's this DNA identification pathway where excellent data can be generated by the labs, but interpretation doesn't happen, or it happens at a much reduced level. And with computers, all the data can be used, information can be preserved, and so on. And then all evidence can be interpreted with match statistics associated. Now, this problem isn't unique to Pennsylvania. It's pretty much national, if not worldwide. And we made some estimates with groups that we work with in New York, Virginia, and so on, asking how many cases are not getting reported out because they have mixtures. So we counted starting 4 years ago when the new federal guidelines came out from the Scientific Working Group on DNA Analysis Methods, or it's called SWGDAM. In fact, the introduction of stochastic thresholds was called SWGDAM 2010. It was the new guidelines that came out in 2010. We asked how many cases are being lost. We estimated from Virginia, remember that statistic that there were 275 cases that were sent back? If you multiply that by 4 years and divide the population of the US by the population of Virginia, you end up with an estimate of about 60,000 inconclusives. Not even low statistics, but inconclusives. If you take a look at New York, they made an estimate that there were about 20 inconclusives a week that were, because there were mixtures, that were not reporting anything out, where their TrueAllele system would have reported something out. And so that's about 1,000 cases a year. You multiply it up again to the whole country, and you end up with over about 160,000 cases. So as a rough estimate, there's about 100,000 cases over the last 4 years across the country where the DNA is not being used, even though it probably is quite informative. And so the estimate is that there's about 100,000 cases where there's DNA evidence data where it's been collected, analyzed by the lab, paid for by the taxpayers, and so on, but not used. Now, you can take this as just being, well, DNA is normal, but we don't, really. And I don't think you would either as investigators. Why is this the case? Well, I'll tell you why I think it's the case and why some of the crime lab directors that I speak with a lot think it's the case. A lot of you are in the CIA unit, and your focus is on information, right? That's the lifeblood of what you do. You want investigative information. When a case goes to court, the prosecution or the defense, and certainly the jury and the judge, are relying on information. What is the evidence and what is the information? And so a large part of the criminal justice system is driven by evidence and information. You're trying to gather evidence that can be used as information by the court, prosecutors, if they want to make sure they have the right person or get a conviction against the right person, they don't have the wrong person. It's all about information, multiple items of evidence, and how informative they are. And the crime labs around the world tend to not work in a culture of information. It's different. They have different constraints on them. They're very much regulated by federal policies. They're audited repeatedly. There's certifications. There's rules to follow. The rules come down from Washington. And what they need to do is to make sure that they stay in business. They need to make sure that they follow protocols, that they follow rules, that the procedures that they have will let them pass their audits and stay in business. And that when they finally go to court and testify, they're testifying about methods that they use, that the FBI uses, that are part of whatever the community is using, that they're very comfortable using. Their main goal is not to provide information. It's to develop evidence and provide testimony. And that's not meant as a criticism. That's the environment that they work in. That's how they stay in business. But a consequence of that is there's a disconnect between investigators and prosecutors and virtually all the DNA labs in this country. And that is the purpose of the DNA lab, is to follow procedures, stay in business, and present evidence in court that they're comfortable presenting about. And that's not always the need of investigators or prosecutors. So whereas you're very much living as investigators in an information 21st century society, the crime labs are not focused on information. That's not what their incentives and what their survival depends on. It depends more on rule following in a way where they can provide information, but it's not the core focus of what they do. And I know some people who are thinking of writing PhD theses about this. It's fascinating, but there is a disconnect between what you might expect a crime lab to produce as information and what a crime lab is expected to produce by the regulatory agencies that let them stay in business. There's a very real disconnect there. On the other hand, there is a problem that there are about 100,000 cases over the last 4 years with complex DNA mixture evidence that could have provided information to society. This extends to DNA databases. The original DNA database was created in the United Kingdom, England. It's the National DNA Database. We've looked at it, we've studied it, we work with investigators there. And it was designed for single source samples. So let's go through this picture of what I know you all know what a DNA database is, but I want to explain to you why everything isn't going up to CODIS, which you might have wondered about. So here, the DNA databases are phenomenal. The notion is you have evidence, you have references. You have criminals, and under the legislation, which tends to always be expanding, first it's, in fact, one state started it with only people on death row, but that didn't make too much sense. But then it starts with felons, and then can they move to arrestees, like in our state. But you have people who could be a likely suspect. Their genotypes are uploaded onto a genotype database, and they're stored. You then get crime scene evidence, which historically, 15 years ago, was single source data from one person. That can be inferred and uploaded to a genotype database. Now, you don't have any suspects yet, but through the magic of computers and databases, the genotypes on the database are all compared with the genotypes from evidence to reference, and a link is made from a crime scene up to the database. There's a match to a person, and then now you can find a suspect. That's the way it's supposed to work. But old-style databases do have issues from back in the old 20th century. They only upload 10% of the mixtures, 10 to 20%. It's about 10% now. And why? Well, the reason is that these database hits are not made using match statistics or likelihood ratios or math. They're just made by association. Some alleles might overlap with some other alleles. And the result is the comparisons between mixture evidence and other crime scenes and with potential suspects can lead to false hits. That's just the way it was designed 15 years ago in CODIS. They don't use math. They don't use genotypes. They just have these alleles up there. And so the result is that there are rules at the national and sometimes the state level that say, well, we don't want to find false hits, and our database can't stop the false hits from happening because that's how it's set up. So we'll block the upload of mixtures by only letting mixtures that have a match statistic of maybe at the level of 10 million, the size of the database, about 11 million people on the National DNA Database now, and just not let those mixtures up there at all, and we'll solve the false hit problem. And that can help police. You're not going to get 10 names or 50 names that are wrong, but it creates another problem that the evidence is never getting up to the database. And if the evidence doesn't get up to the database with mixtures, if 90% of the mixtures can't be uploaded because they're using alleles and lists of alleles based on these inclusion methods and not genotypes, then the evidence can't get up there, the comparisons can't be made, and you're blocked from actually using the evidence that you've collected to find criminals. TrueAllele is a genotype database, and so it's based on all the principles that you saw. There's a TrueAllele database that's sort of packaged in with every TrueAllele system, but there are some groups that have expanded this to a much larger scale database. And there, everything's based on genotypes with probability. Why? Because that's what preserves the identification information. These lists of alleles, as you've seen in the different studies and cases, don't preserve information. You either get lower match statistics or inconclusives. But if you can upload all the mixture evidence because it's based on genotypes, and the comparisons and the hits are based on real statistics, then from our studies that we and other groups have done, you find two things. First, the database method is sensitive. It will find the true hits. You will get that this piece of evidence, just like in a case, but now on a database, this item of evidence from a mixture is identified with a particular individual who's a convicted offender with a match statistic, say the number is, 1 million. You're getting real match statistics from the information from the genotypes. But it's also specific. What our studies show is that you don't get false hits. You saw that in the picture I showed you from the Kern County case. We do that study not just in validation studies. We tend to report that in the discovery materials for every case that we go to court on. We show the huge separation between comparisons of evidence with random people and the actual suspect. You rarely are crossing a level of 1 or 10 as a match statistic. You're not getting the sort of false hits that CODIS finds. And since you're finding the true hits and it's specific, you're not getting the false hits, everything from the crime lab that's a mixture can go onto the database. You can't do this with CODIS at any level, LDIS, SDIS, or NDIS, but you can do it with a genotype database where all the mixtures can be interpreted, the genotypes and their probabilities can be uploaded, the matches can be made, and you don't get false positives, and so you don't need to restrict what's going up there. You don't need to say only the 10% of the easy mixtures go up. 100% can go up, and that way you have all the evidence being matched against all the people or being compared, and when there's a match, it's something you can rely on. So that's another impact that happens when you move from these lists of alleles and inclusion methods that don't preserve information very well to genotypes using more modern computing that have as a goal preserving identification information.